Hey everyone, welcome back to my channel Synapse. I am Dia, a third year medical student, and today we are diving into a crucial pathology topic, that is necrosis. What happens when cells die? How does the body respond? Stick around and let's break it all down. Necrosis. Now, it is defined as a localized area of death of a tissue, which is followed later by the degradation of the tissue by the hydrolytic enzymes. Now, some enzymes named hydrolytic enzymes are liberated from the dead cells, which cause the death of the tissue. Now, an important thing to remember is that necrosis is always accompanied by the inflammatory reaction. Now, you'll ask why? Because the body recognizes these dead cells as harmful and sends immune cells to clean up the mess. Now, one key point to understand is necrosis is accidental, whereas the apoptosis is basically the programmed cell death. Okay, which is a controlled process. Now, necrosis is unregulated and it is always pathological. That means it is never a normal process in the body, it always happens due to injury or any infection or toxins or other damaging factors. Now, another thing to note is that necrosis doesn't affect just one cell. It involves multiple cells in a given area. Now, we'll talk about the mechanism. Necrosis occurs in two ways, either by denaturation of the proteins or enzymatic digestion of the cells. Now, take a look at this figure. It demonstrates necrosis of a cell. Now, notice how the contents leak everywhere, which is triggering inflammation and potentially leading to organ failure. Now, what happens in a normal cell? Necrosis is caused by both external and internal injuries. As a result, the cells swell and burst. It bursts down so that the contents release out. Leakage and enzymatic digestion affect neighboring cellular contents. Now, here is a summary of all that it damages the plasma membrane, there's a leakage of the cellular contents, and increased inflammatory infiltrate, which is there to clean the debris. Morphologically, necrotic cells show distinct changes. First, they appear densely eosinophilic due to loss of the cytoplasmic RNA. This means that they stain more pink under the microscope. Next, they take on the more shiny appearance, which is due to the loss of glycogen from the cytoplasm, which alters the cell's texture. Another key feature is the moth-eaten appearance. This happens because lysosomes digest the organelles, leaving vacuolated spaces in the cytoplasm. Now, necrosis is classified into different types based on its appearance and the underlying cause. The main types are coagulative, liquefactive, caseous, fat, and fibrinoid necrosis. Let's start with coagulative necrosis. In coagulative necrosis, the architectural outlines of the cells are preserved, but the cellular and the nuclear details are completely lost. This gives the tissue a ghost-like appearance or a tombstone appearance under the microscope. Now, where do we see this type of necrosis? It is actually the most common type of necrosis. Okay, so the main causes include ischemia due to thrombosis or embolism. It occurs in all organs except the brain. Next is the mild burns, also known as a thermal injury. In Zenker's degeneration necrosis, which is a specific type seen in muscles. Coagulative necrosis occurs in infarcts of solid organs except the brain. The most common site is the heart, but it can also occur in liver, kidney, and spleen. Now let's talk about its gross appearance. The affected site is pale, firm, and slightly swollen. In this figure, you can see the gross appearance of the necrotic kidney. Notice the yellow portion, which indicates the coagulative necrosis. Under the microscope, the hallmark feature is the conversion of the normal cells into tombstones, meaning the cell outlines remain but nuclei are lost. In this histological slide of the kidney, you'll see different structures marked. The green star shows healthy cells, which is less pink and visible nuclei. The blue star marks Bowman's capsule. The yellow star highlights the necrotic portion. Notice how the cell structure is still present, but there is no nuclei present. in this region. The next, after coagulative necrosis, we'll discuss about the liquefactive necrosis. In liquefactive necrosis, the architectural, cytoplasmic, and the nuclear details are lost completely. The dead tissue transforms into a semi-fluid material due to the degradation of the hydrolytic enzymes. Now, this type of necrosis is also known as coagulative necrosis. Now, what causes liquefactive necrosis? The main causes include pyogenic bacterial infections. These lead to pus formation. Ischemic necrosis of the brain. Unlike other organs, the brain undergoes liquefactive necrosis due to high lipid content. Now, the common examples of liquefactive necrosis include the brain infarcts, the fungal infection, and the abscess formation. Grossly, the liquefactive necrosis appears as liquefied center containing necrotic debris with a cyst wall. This happens because the dead tissue is completely digested by the hydrolytic enzymes, as studied previously. In this figure, you can see an example of liquefactive necrosis in the brain. Notice how the tissue has softened and broken down in this region. This is important because coagulative necrosis does not occur in the nervous system. Instead, the brain undergoes liquefactive necrosis due to high lipid content. Now let's look at the microscopic feature. The necrotic area appears as a cavity filled with necrotic neutrophils, bacteria, and tissue debris. This is surrounded by a cyst wall which is made up of proliferating capillaries and inflammatory cells. Here's a histology slide of liquefactive necrosis. Notice how the middle area is pinker, while with more spaces and fewer neurons. If we zoom in, we would see macrophages filled with lipids and debris, clearing the necrotic material. Caseous necrosis is a form of a cell death where the architectural and the cytoplasm and the nuclear details are completely lost, leaving behind amorphous and a granular debris that looks like cotton cheese, hence the name is caseous. The most common example is Mycobacterium tuberculosis, but it can also be seen in syphilis, histoplasmosis, and coccidiomycosis, all of which trigger a granulomatous inflammatory response. Now, if we look grossly, caseous necrosis appears soft, granular, and yellowish, which resembles dry cheese. This appearance is due to the lipid-rich cell wall of the mycobacterium and the fungal infection, which resists the complete digestion of the immune cells. In this figure, you can see the lung containing caseous necrosis due to tuberculosis. Notice the yellowish, white, and cheesy debris. This type of necrosis is commonly seen in granulomas, which is a collection of activated macrophages and surrounded by lymphocytes, often forming a central area of caseous necrosis. Now let's look at the histology of the caseous necrosis. If we examine the tissue section under the microscope, we'll see the center of the necrotic area is made up of the structureless eosinophilic material having a scattered granular debris of disintegrated nuclei. This gives a homogeneous pink appearance on the H&E staining. Now in tuberculosis specifically, caseous necrosis is typically seen within a granuloma. A granuloma is a collection of immune cells, mainly activated macrophages, also called as epithelioid cells. Now these epithelioid cells are slipper shaped. So they surround the caseous necrosis. And around these activated macrophages is the lymphocytes. So this forms the granuloma. The periphery consists of the immune cell attempting to contain the infection, whereas the center contains the necrotic debris. Now the fourth type of necrosis is the fat necrosis. It is a special form of cell death which occurs at fat-rich anatomical locations in the body. It refers to the death of the fat cells in a living organism. This type of necrosis is seen in conditions that involve trauma, inflammation, or enzymatic digestion of the fat tissue. Fat necrosis can occur due to different causes depending on the location. Firstly, it is the pancreatic fat necrosis, which occurs in acute pancreatitis due to the release of the lipase and other pancreatic enzymes, which break down the fat. Traumatic fat necrosis is seen in breast, often due to injury or surgery, leading to localized fat destruction. Fat necrosis in the mesentery occurs due to inflammation, commonly associated with conditions like peritonitis, or ischemia. Grossly, when we see yellowish white firm deposits within the fat tissue. These deposits form because of the fat saponification, where the released fatty acids combine with calcium to form the chalky white deposits. Now let's look at the histology of the fat necrosis. Under the microscope, we see the necrosed fat cells which take on a cloudy appearance which is surrounded by the inflammatory reaction. This type of inflammation is a response to the release of fatty acids from the damaged fat cells. If we examine the fat necrosis adjacent to the pancreas, we notice two distinct areas. On the left side, we see normal fat cells or steatocytes which have clear cytoplasm and distinct nuclei. This is marked by green star. On the right side, it's the necrosed fat cell appears vague and ghost-like, meaning there is cellular content, the cellular outlines are blurred. You can't see the proper cellular outlines. These cells have lost their peripheral nuclei. And their cytoplasm has turned into pink, amorphous mass of necrotic material. This area is marked by yellow star. Fibrinoid necrosis is a special type of necrosis characterized by the deposition of fibrin-like material in tissues, particularly in the walls of the blood vessel. This type of necrosis is commonly associated with immune-mediated vascular damage. The main cause of fibrinoid necrosis is the immunological injury of the vessel wall. This occurs in conditions like vasculitis, malignant hypertension, and autoimmune diseases like SLE. Under the microscope, the affected vessel wall shows a bright eosinophilic, hyaline-like deposition in the vessel wall, which is surrounded by the nuclear debris of neutrophils. If you look at this figure, we can observe an artery which is surrounded by a bright ring of necrosis. This bright eosinophilic ring represents fibrinoid material. Additionally, there is inflammation present, confirming that the necrosis is associated with the immune-mediated vessel injury. And that's a wrap on today's video on necrosis. If you found this video helpful, give it a thumbs up, subscribe to Synapse for more medical content, and drop a comment below with the topic you want me to cover next. Until next time, keep learning, stay curious, and I'll see you in the next video.